Per is an ornithology professor, an author, and has been a keen birder since early childhood. He is widely traveled and the author of more than 100 scientific publications and dozens of popular science articles, including many on difficult to identify bird species. He's the first author of two highly acclaimed books, A Field Guide to the Rare Birds of Britain and Europe, and The Pipits and Wagtails of Europe, Asia, and North America. And he's also painted those pipits as well. He's also the co-author of the two volumes of Handbook of Birds of the World and the 6 chapters of the largest avian radiation, The Evolution of Perching Birds, or the order Passiformes. Per has been involved in descriptions of 7 species and 3 subspecies of birds new to science. He's an advisor to the IOC World Bird List, a former member of WGAC, or the Working Group of Avian Checklists, which is now called AVILIST, and he's the deputy editor-in-chief of the International Ornithological Journal, Avian Research. So thank you for joining me. That's quite an impressive resume. We really are so glad to have you here to share your expertise.
So thank you very much. It's a great pleasure and a great honor to be here tonight. And I'll be talking to you about integrative taxonomy. So I'll start by explaining some of the terms that I'll be using tonight. Then I'll talk very little about species concepts and general problems with delimitation of species. I'll talk about the increase in the number of species recognized. I'll touch very briefly and superficially on speciation. Then I get to integrative taxonomy, and I will explain what that is, and I will give some examples, mainly from research that I've been involved with. Finally, I'll just talk a little bit about computer-based taxonomy, which is something that has become popular in recent years. So my main conclusions here is that taxonomy is basically the basis for all other biological research and biological conservation. And the number of species that we recognize in the world has increased quite dramatically in recent decades as a result of our increased knowledge about these birds. And I would argue that species delimitation is often unavoidably subjective, and that is because of the gradual, uneven process by which new species are formed. And I like to see species as hypotheses, and as such, they can be revised when new data come to light. And what then is integrative taxonomy? Well, it's the integration of multiple complementary lines of evidence, and I think that's the best practice. There are some pitfalls with DNA analyses and also with computer-based species delimitation, which I'll come to a bit later. So let's start to look at a phylogenetic tree, because they are important for taxonomy and systematics. Here we've got 5 different groups, A, B, C, D, E, which are indicated by differently colored branches. So the way to understand these relationships is to look at the nodes here indicated by arrows. So these nodes, they represent a hypothetical most recent common ancestor between the groups at the branching points here. So if we look at, for example, B and C, they are more closely related to each other than to any other group in this tree. And similarly, A is more closely related to B and C than to any of the others. But if you look in this way, vertically, C and D are adjacent to each other, but they're not each other's closest relatives, because in order to find their most recent common ancestor, you need to go down to this node here. So you need to follow the branches all the way down here and then up to here. So you see, they are not each other's closest relatives. So here you've got other ways of drawing these trees. As you can see here, the topology, the relationships here are identical to the first tree. It's just different ways of drawing them, because at each node here, you can spin the branches around. So you can get lots of different ways to draw these trees. Here is two other representatives. And if you look at the one to the right here at the bottom, you can see that the branches here have unequal lengths. And the branch length in this case, unlike in the other trees shown here, are representing the level of divergence, the level of differentiation along these branches. So in this case, you can think of this as time. So you've got a time line here. And here's two other ways of drawing trees. So a few terms that are important. A taxon, plural taxa. That's any named taxonomic group. It could be species, could be subspecies, family, order, or some other level. And sister species, sister families, sister clades, that is the closest relatives. So for example, these two here, the closest relatives. And a clade then, what is that? It's the same thing as a monophyletic group, which means the most recent common ancestor plus all and only all of the descendants. So here is one example of a monophyletic group. And here's another example. So you see the first example I gave had a group with only 8 species. And here we've got 3 subclades with 5, 3, and 3 species. Here's another example of a monophyletic group. And here's another one, and here. So monophyletic groups, they form the basis for modern classifications. And if we look at these 3 tits here, they were previously all placed in the genus Parus. But as you can see here on this phylogeny of all the different genera of tits as they are defined today, you see there's lots of different names, and Parus is only here with 5 species in this little clade here. So why is that? Why have all these names appeared instead of only Parus? Well, it's because of this guy here. This bird, which breeds on the Tibetan plateau, where it's usually, well, no trees or very few trees. It nests on the ground in holes, and it hops around like a little kangaroo on the ground. It was previously thought to be a corvid, so a relative of crows, and it was only few decades ago that Helen James and Per Eriksson, Fu-Ming Lei, and a few others realized that this is not a corvid, it's a tit. And as you can see on the phylogeny here, it's actually more closely related to the great tit than great tit to blue tits or to this marsh tit down here. And the reason why the taxonomists who were revising the taxonomy here, they really wanted to highlight the distinctness of this bird. They wanted to keep the name Pseudopodosis, by which it was known. And then all these other names had to be used, because if Pseudopodosis would be used, the old traditional Parus would no longer be monophyletic. And that is not acceptable according to modern classification. As you can also see here, circumscriptions of genera, families, and other levels is very subjective. We saw that there are many different possible monophyletic groups, and in theory, any of these monophyletic groups could be treated as a genus. With regard to geographical distributions, allopatry or allopatric means that you have separated geographical distributions. Parapatry, then you've got abutting distributions. They could be marginal sympatric. Different authors use the term slightly differently. But sympatry, that requires some sort of geographical overlap. And of course, you can imagine all different transitions between these levels. You can have wide allopatry, or you can have narrow allopatry approaching parapatry, and then you can have wide symmetry, or just very narrow marginal symmetry. Species concept and species criteria important. Now, a species concept tells you what species are. So that is ideas of what species are. Species criteria, on the other hand, they describe how species may be delimited in practice. So what then is a species? That's the eternal question. And here I've brought together three species concepts, which I refer to as layman species concepts. So Encyclopedia Britannica at the top, it says species are related organisms that share common characteristics and are capable of interbreeding. The Free Dictionary says a group of closely related organisms that are very similar to each other and are usually capable of interbreeding and producing fertile offspring. And finally, Wikipedia, a population of organisms in which any two individuals of the appropriate sexes or mating types can produce fertile offspring, typically by sexual reproduction. Well, these are all necessary properties of species, but they're not sufficient. So general comment here is that many species that everybody recognizes to be different species are actually capable of interbreeding. And many of these actually produce fertile hybrids. But this is really important. Different species usually do not mate with each other. And here's an example of 4 ducks that have hybridized, and fertile hybrids have been observed. And in fact, around 15% of all bird species are known to have hybridized with at least one other species in the wild. In captivity, the figure is even higher. Ernst Mayr has formulated the most influential species concept, at least in ornithology. And according to him, species are actually or potentially interbreeding populations, which are reproductively isolated from each other by intrinsic barriers. So intrinsic barriers means not purely geographical barriers. And Ernst Mayr, he recognized species as well as subspecies. Joel Craycraft, he formulated one of the many phylogenetic species concepts, often abbreviated PSC, as smallest group, diagnosably different from others, and then descended from a common ancestor. So diagnosably different means that if you look at some feature, could be plumage, could be some other features, they need to be identifiable. They need to have some unique features or combination of features. And only species are recognized according to Craycraft. So no subspecies there. And in reality, his version of the phylogenetic species concept is in reality very similar to morphological or phonetic species concept. And there are many, many, many more species concepts that have been proposed by different people. So then what is a species? Well, if we're in a certain spot, such as Eastern China here, we usually don't have a problem delimiting species because the species that occur there during the breeding season, I should add, they look different, they sound differently, they are usually ecologically segregated in some ways or another, and importantly, they don't interbreed. So we don't have any problem with those. But when we have allopatric breeding areas or parapatric ones, we often have problems, such as these Iberian magpie and azure-winged magpie. They were previously treated as the same species. But when you have this situation, you must decide whether a certain taxon is a different species or a subspecies of the same species under the biological species concept, or whether they are actually different species under the phylogenetic species concept. So here we've got 2 different birds which look differently, they sound differently, they differ in size, shape, plumage, behavior, They are not each other's closest relatives, they're not sisters, and they occur together, they don't interbreed. So there's no doubt about this. They are different species under any species concept. But what about these guys? They are parapatrically distributed. This blue tit occurs in Europe and Western Asia, whereas these guys here occur in North Africa and the Canary Islands. And they differ in structure, in plumage, they've got different vocalizations, and there's a deep genetic divergence, as you can see here. So even though there is some differentiation between North African and Canary Island birds, as you can see here, there's very, very deep divergence between European birds and birds from North Africa and the Canary Islands. You can see that on this little inset here, but it's the same one as here. So there's deep divergence between those. So based on that, yeah, there should be different species, but then we've got a complication here. We've got the azure tit, which is actually based on genetics, more closely related to European blowtits than European blowtits to North African and Canary Island blowtits. And even more complications when we take into account that the azure tit is actually partly sympatric with the northern blue-tits, and they do hybridize where they meet. And here's a study that was published 2 years ago, where Martin Irestedt and colleagues looked at blue-tits and azure tits and hybrids. And they found, of course, some pure blue-tits and some pure azure tits and all different levels of hybridization between them, as expected, based on what we knew before. But here's interesting that 3 of their individuals actually looked identical to azure tit, but they had a certain level of intermediacy between blue tit and azure tit in their genomes. Here's an example where we have 3 different geese that look slightly differently, and they breed along the Arctic coasts, as you can see here, and different populations that are usually treated as different subspecies have different migratory routes. So Bernicla, which breeds in Western Siberia, winters in Western Europe. Chrota, which breeds in the northeastern part here, northeastern US and Greenland, has 2 different wintering populations, one on the east coast of the US and one in Ireland. Whereas nigricans from eastern Russia and western North America winters on both sides of the Pacific. So there is some sort of migratory divide here. And at this place, at the Oleniec River, east of the Taimyr Peninsula, there's been a study where it was found that at the same time, Bernicla was sitting on eggs while nigricans had actually chicks. And since these birds mate on their wintering grounds, they arrive at the breeding grounds from different directions and at slightly different times. So the level of introgression, the level of interbreeding between them could actually be rather slight. The situation here, I think, is slightly less well known, but it's something that's interesting to study and shows the complexity of taxonomy. The number of bird species in the world has increased quite a lot from Linnaeus, or Carl von Linné, as we call him in Sweden. He was aware of 554 species of birds in the world. Then in 1900, early 1900, Sharp, who was working at the British Museum of Natural History, as it was called in those days, he identified no less than 18,900 species. And that was based on a so-called morphological species concept. So he just looked at the specimens and identified everything that he thought was distinctive as a species. Then in 1951, Ernst Mayr and Amadon published a list based on Ernst Mayr's biological species concept, which lumped many of Sharp's species into polytypic species, so species with multiple subspecies. And as you can see here, the number dropped quite dramatically, almost 10,000 species fewer than Sharp. Number went up a bit, and I have a list which will be published soon, will recognize 11,136 species. So there's been quite a dramatic increase, as you can see here, since Ernst Mayr published his list. And actually, Barraclough et al. Published a paper in 2016 based on a phylogenetic species concept where they estimated the number of species to approximately 18,000, which is, as you can see, pretty similar to Sharp's estimate. So there are in the world today 4 major global checklists. That's the Clements-Ebert checklist, that's the IUC checklist, BirdLife, and Howard and Moore. Howard and Moore was published a bit earlier. It's been less updated than the others. But between these, there is no less than between 1,200 and 2,000 incongruent cases. So cases where at least one of the checklists differs from the others in species limits or in genus. So there will be a list called ABLIST, which is a unified global list published soon. And then the question is, why has the number of species increased so much? Well, it is because we have learned so much more about birds. We got much better tools these days than Linnaeus here. We got amazing binoculars, telescopes, fantastic cameras. We got excellent recording equipment. We can analyze DNA, and the world is actually quite open to us. We can travel almost anywhere these days. So we have learned so much more. The old taxonomists, they would be limited to specimens that either them themselves collected or others collected for them. They would sit down in a museum collection and they would compare these. And they would come to their conclusions based on plumage and structure, because that's what they could study those days. Now we do the same. We still study plumage and structure, but we can also study vocalizations much better. We can study sexual behaviors and other behaviors that we think might be important. We can look at the bird's ecology. We have often quite got good information on their geographical distributions, maybe whether they come into contact. We can study hybrid zones, etc. And of course, we got access to DNA. So during the period from Ernst Mayr's publication in 1951 to 2024, only approximately 300 species new to science have been described. So that means the main increase is due to subspecies being elevated to the rank of species. Okay, so that's good, but why on earth are there so many uncertainties? I mentioned between 1,200 and 2,000 in this ABLIST adventure. Let's have a look at speciation, just to try and understand this. We start by a single ancestral population, and this could potentially become subdivided into 2 or more geographically isolated populations. With time, these will inevitably diverge. They will diverge in plumage, in structure, in vocalizations, ecology, genetics, etc. But it's important to remember that the rate of change can be very unequal. So, as you all know, in some species, plumages don't differ very much between different species, whereas vocalizations might be quite distinctive. In other species, it's just the opposite. Voice has not changed much, but plumages or structure might be much more different. In case these populations expand their ranges and come back into contact again, we call that secondary contact, then it's quite possible that reproductive isolation has evolved. And reproductive isolation means that either they don't mate with each other, they don't interbreed, we call that premating barriers, or if hybrids are formed, these are less fertile or less viable, and then we refer to post-mating barriers. Alternatively, there could be extensive hybridization, there could be a hybrid zone formed, and possibly they could just merge all together. One important thing is that gene trees actually differ from species trees. And here is an example where we have some of the possible trees for 4 different wagtail species. And this particular tree here was found in the majority of the genetic regions that were analyzed. Whereas this tree, which has a different relationship, different topology, it was found in almost the same number of genetic regions. And this one also fairly similar, this one here. And then you see there's a diminishing number of regions supporting other trees. So one of the reasons why a gene tree can differ from a species tree which I would refer to as the phylogeny, is because of what we call incomplete lineage sorting. It's often just called ILS. And we've got here a species phylogeny with 3 different species. Here it starts out as 1 species. Then we've got a speciation event. So here we've got 2 species, and then another speciation event giving rise to 3 different species. And the colored branches inside here is a gene, one of the many, many, many genes that is shown here. And the different colors, they represent different mutations along these genes, giving rise to slight differences. So if we move back a little bit in time, if we say that we are at this point in time and look at the species here, we call them A, B, and C, you can see that the relationships depend on which genes we sample here, which alleles we sample here. So if by chance we get this X gene, and we get this here and one of these, we have the same relationship as in the species phylogeny, as you can see here. But if instead we sample the Y gene, you see we get a relationship that is incongruent with the species phylogeny, because then B and C are inferred to be each other's closest relatives. If instead we look at today, and we call these what is this in English? A prime, I think, B prime, C prime. So if we look at these 3 species here, and we infer, you see, well, note first here that this allele here, it's gone extinct. It no longer exists in this population. So this is the only one that exists. And then, as we saw before, we get the wrong relationships based on this particular gene. So in this case, this Y lineage here, it misrepresents the species phylogeny. Then, of course, we could have introgression. So that means we have gene transfer because of interbreeding between these 2, and that will further model the situation. Then one further complication is that maybe these two, as shown here, will interbreed so much that they will actually merge at some point in time and become a new lineage here. So in that case, evolution is more like a network rather than a bifurcating tree. And this is probably quite common in nature. So the problem why we have problems the reason why we have problems delimiting species is because speciation is a gradual process. And if we look here at this simplified situation where we have one species that splits nicely into two at this point here, with time is along here, as we know, there could be, at this point here in time, there could be plumage differences that have diverged so much that they are unique to each of these lineages. So they are diagnosably different by plumage. At this point in time, maybe the vocal differences are significant and diagnostic. At this point in time, maybe they have evolved reproductive isolation. Here, they have become finally monophyletic. So in this zone here, are we talking about one or two species? At this point in time, they are so different that everybody will agree that these are definitely two different species. And this is the basis for the so-called general lineage concept, which was formulated by Kevin de Queiroz, where he concludes that these properties here represent different stages in the speciation process, and they are variably important for different species concepts. So these could be used for the phylogenetic species concept, sensu craycraft, and these could be used for another phylogenetic species concept, whereas here we've got the hallmark of the biological species concept. So importantly, a continuum as this is can only be subjectively divided, and therefore species delimitation is often simply inherently subjective. And therefore, I would argue that most of the controversial taxonomy cases where taxonomy is arguing, is it a species? No, it's a subspecies. Most of those cases are actually here in this gray zone. And also many of these cases are very poorly known. We haven't got any information on the vocalizations or their interactions with other species, etc. So species, in my view, are testable hypotheses, and therefore we have to accept that when we get more data, we might change those hypotheses, we might revise them. So the best solution then, I think, is what we call integrative taxonomy. And I would recommend that irrespective of your favored species concept. What does it do then? Well, it integrates multiple independent lines of evidence of lineage separation under the phylogenetic species concept or the general lineage concept, or of reproductive isolation under the biological species concept. As we saw on the previous slide, reproductive isolation can also be used to identify a lineage. But size, structure, plumage, important. Songs, calls, important. Sexual displays could be, for example, song flights in larks, very important. Ecological differences can also be obvious between different species. And of course, genetic differences. And when it comes to genetics, one could look at trees like here, one could look at branch lengths. And I do actually think that it's generally more important to look at relative branch lengths than actual time estimates, because time estimates are, well, I generally don't trust them. And of course, there are problems with relative branch length as well. If there's hybridization, for example, that will affect branch length. So, but anyway, trees can be important. Same with this type of graph, which is an admixture plot, where you've got different groups identified here, and you can see that some of them are less well-defined. You can have a PCA plot, where you identify different groups, different clusters of individuals based on their genetics. Then, of course, geographical distributions, very, very important. And also interactions. If there is geographic contact, interactions, super important. Importantly, trait divergence is heterogeneous. Remember that. And therefore, species hypotheses aren't falsified by the absence of a certain species property, for example, plumage differences, if they are supported by other properties. So even if you have very, very similar plumages, if they differ in lots of other aspects, they could still be treated as species. And the more support, the better. And independent support is better than additional support of the same class. So it's better to have vocalizations and morphology rather than 2-3 different classes of morphology only. But one should try to get as much as possible. If we start looking at allopatric distributions, morphology, vocalizations, behaviors, ecology, everything, we take that into account. We may want to have some sort of yardstick. We might want to compare with other species that we know, for example, are reproductively isolated. And we want to compare the divergence in those different aspects that we're looking at with the other species that we know for sure are reproductively isolated. Joe Tobias and co-authors, they proposed a way of quantifying and comparing species using what has also been called the 7-point rule, and which is supported by BirdLife. And that gives scores to different phenotypic traits, so plumage, structure, vocalizations. And then if those sum up to 7 or above, they are treated as different species. I will not go into detail about this, but personally, I think it's a very artificial way of delimiting species. Do remember, never forget, trait divergence is equal. And DNA can be tricky. We'll look at that in a little while. When we have a situation where we have sympatry or parapatry, so species are either just right adjacent to each other or have overlapping distributions, we'll look at the same things. And if they are distinct, that indicates there's no gene flow or very, very limited gene flow. And then, of course, they are different species. No problem. We could also look at interactions. We could, for example, do playback tests, although I want to stress that they should be interpreted very cautiously, because you could have cases where different species actually do respond to each other's vocalizations because they are competing for some reason. You could also have cases where there's no response, even though some other time they would respond. And then, of course, what we do test usually is only male interactions. It's very, very hard to get females to respond. If we have a hybrid zone, what's the width? Does it change in width? Does it move? Does it change location? What's the proportion of hybrids in the zone? Does there seem to be sort of free interbreeding, or is there just a small proportion of the individuals in this hybrid zone that are actually hybrids? So here, DNA can be tricky, as I said. Let's look at these wagtails, amazing birds, distributed widely across the Palearctic. And as you can see here, there are lots of different taxa that differ mainly in one single plumage trait. That is the head pattern of adult males in summer plumage, in breeding plumage. Then there are some differences in vocalizations between different regions. And these days, usually the eastern birds, 3 of these subspecies, Chuchensis plexa and Taiwana, and Macronix 4, are treated as eastern yellow wagtail as a separate species from the western birds here, with all these other subspecies, western yellow wagtail. But importantly, even though you can see there's a lot of differences in head pattern, you see you've got birds with a yellow supercilium and gray crown here. You've also got a population here. You've got one here. You've got birds with all gray head here and here and here and here. Birds with white supercilia in different places as well. So all these, where their ranges come into contact, they do interbreed to varying levels, but nevertheless, they do interbreed. Even here, between what is now treated as 2 different species, there is interbreeding. And then we've got this citrine wagtail, which differs in multiple plumage aspects in all plumages, so not only adult male breeding. You can see it's not only the head pattern that is distinctive, it's also got this neck pattern and color of the upper parts and wing bars. So lots of differences, and also females and young birds differ. And as you can see here to the right, citrine is more similar to western yellow wagtail in its song. It's more similar to eastern yellow wagtail in its call, also in its hind claw length. When it comes to sexual dichromatism, so that is the difference between male and female, it's more similar to the western again. And first winter plumage bears more similarity to eastern yellow wagtail. But importantly, citrine wagtail has a wide overlap between with the others. So it's sympatric with both western and eastern yellow wagtails, and usually it does not interbreed. Interestingly, there don't seem to be any apparent ecological differences. I've noticed that in only one place in Mongolia, but otherwise they can breed right next to each other. And normally they do not hybridize. So in this case, these 3 lineages, eastern yellow wagtails, western yellow wagtail, and citrine wagtail, they seem to be approximately equally old, so of the similar age. So it's quite clear that in this case, you've had very unequal rates of plumage divergence. Citrine wagtail has diverged much more than all the others. If we look at the genetics, you can see that the mitochondrial tree here is very, very odd, because you've got the citrine wagtail in 3 different places, here, here, and here. And then you've got western yellow wagtails here, and you've got eastern yellow wagtails in 2 positions. And even more strange, you've got the grey wagtail and the African pied wagtail also here as more closely related to these. If we look at a tree based on genomic data, so nuclear DNA, a species tree, you see that the African pied wagtail, which is here, is now in this group together with all the other black and white species. And citrine wagtail forms one clade, all of them, and also the yellow wagtails form a monophyletic group. So in this case, it's quite obvious that something has happened to the mitochondrial tree. It does not reflect the species phylogeny. Here's another example where it seems quite likely that there has been introgression, so that is transfer of mitochondrial DNA between two different populations of Godlewski's bunting, which is the green distribution here. And you've got northern birds here, you've got southern birds here. If you look at the mitochondrial tree, you can see that northern populations of Godlewski's bunting are most closely related to meadow bunting, whereas southern populations of Godlewski's bunting are more closely related to rock bunting. And they've been separated for quite a long period, more than 2.5 million years, according to this tree here. Mitochondrial DNA, important to remember, is inherited on the maternal side, so from mother to daughter. And it's inherited as one single locus. And we've got here the W chromosome, which is unique to female birds. Male birds do not have a W chromosome. So here you can see that this topology, the relationships among those, is the same, which is to be expected, sort of in this case. But if you look at the dates here, you can see that divergence times differ. And as I said before, I don't really trust divergence times very much. But anyway, we got the same phylogeny here. If we look at genomic SNPs, so a SNP is a difference in a gene between different sequences in a gene alignment. So then you can see that the two Godlewski's buntings, they always form one group here, always form one group. And same in a tree based on nuclear DNA, they come together. So in this case, what we concluded is that there's probably been interbreeding between populations here, which has transferred mitochondrial DNA and W chromosome, causing this discrepancy between the two trees, as you can see here, the nuclear tree and the mitochondrial tree. Genetic distances. Beware. You often hear people saying, and you read in papers, that they differ by 5% or they differ by 7% or any other value. This is totally meaningless. Also, cutoff values for species rank is not a good idea at all. As you've seen before, where do you draw the line? As you've seen on the tree that I showed, where do you draw the line? Where do you say these are different species? And the reason why this is pointless is because different loci, so different genes and different models, cannot be directly compared. So if somebody says they differ by 5%, you have to ask, wait a minute, which gene were you looking at? And how long part of that gene did you look at? Did you look at the whole gene or just part of it? If you only looked at part of it, which part did you look at? Because that can also differ between different genes. And which models of gene evolution did you use when you estimated those divergences? So there's lots of different caveats here, and you can read more about those in this paper here. If we look at the leaf warblers, the Phylloskopus warblers, on the Eurasian continent and in Japan, in Peter's checklist, published in 1986, there were 39 species recognized. Gill et al. Last year recognized 62 species. And there's only been 5 new species in this period. So the majority here is the result of taxonomic splits. So as you can see here, in some cases, one species turned into 2 species, and there's even been one species that is now treated as 6 different species. 2 of these were admittedly new, but here you've got a case where 1 turned into 5. So here are the 5 new species that were described in this period. And I would like to take the opportunity to say that this Alström's warbler, although I'm proud of the name, it was not my idea to propose this English name. We did describe this species, Urban Olsson and I, but we did not propose an English name at all. So it was first Frank Rind in a paper in Birding Asia, and then the editors of the Handbook of the Birds of the World, for which I wrote the species account that wanted to use this name. And I had them state in the introduction of the book that it was their choice. It was not at all my proposal. Let's look at the limestone leaf warbler, which is one of those. We started to study it in Vietnam, Laos, and it's extremely similar to the sulfur-breasted warbler, which occurs in China. It's also rather similar to the yellow-vented warbler, which occurs in the eastern Himalayas. But you can see the yellow-vented is more different in plumage because it's got a white belly and flanks, unlike the others, which are very yellow. Now, this is just a highlight here, but they are really bright yellow on the other parts. So this bird that we called Philoxopus calciatelis, it's got a proportionately larger bill and smaller size than the others. And it does differ in vocalizations. So here is calciatelis. And now I'm going to play the sulfur-breasted from China. So there are consistent differences, even though there is a lot of individual variation and every individual has a certain repertoire. But if you analyze lots of these, and that's something I want to stress, when doing sound analysis, it's important to have multiple individuals. And especially if every individual has a repertoire, it's important to have long recordings as well to compare. We'll see if I got a recording of this as well. Yeah. So all these differ in vocalizations, and based on genetics, it turned out that the cantator is more closely related to calciatelis than calciatelis to rickety. So these two look very similar, whereas this one looks pretty different. But these ones are each other's closest relatives based on genetic markers that we used. The Arctic warbler has been split into 3 species: Arctic, sensu stricto, Kamchatka leaf warbler, so Arctic warbler is all the way here, and then Kamchatka leaf warbler, the red markings here, and the Japanese leaf warbler here in blue. They'll look very, very, very similar. The Japanese one, it's got usually slightly more yellowish underparts, but they can be really, really, really similar. Well, I want to modify here. You can see the northernmost island, Hokkaido. There's the Kamchatka leaf warbler occurs there. So based on DNA, these 3 populations have been separated for a long time, between 2 and 2.5 million years. So here is the northern one from Norway all the way to Alaska. Here's the Kamchatka, Sakhalin, and the northernmost Japanese island, Hokkaido. And then here is the rest of the Japanese islands. And they got different vocalizations. I will play first the song of the northern one, the Arctic warbler. It's got a very simple stereotyped song. And I will play another individual, and this is from further east. I can't remember whether this particular recording is from eastern Siberia or Alaska, but it's usually got, or quite often got, these little thin lines at the beginning of each strophe here. It sounds slightly sharper. Still a very simple song. The Kamchatka leaf warbler's got a much more complex song. You can see it's got like a little group of elements that is repeated multiple times. And note again, you've got these very thin elements here at the beginning. Sounds like this. Then the Japanese, it's got an even more complex song, but again, repeating groups of elements. And note that these thin elements are lacking. Sounds like this. Also, the calls differ. So the Arctic, the northern one, like this. And then the Kamchatka warbler, slightly more cracked voice. And the Japanese, low-pitched. So quite different. The lesser short-toed lark, Asian short-toed or sandlark complex, they have been treated as one species, two species, three species, and we propose they are better treated as 4 different species. We collected data from across this range here. So we collected DNA samples. We collected morphology and vocalizations. And in Mongolia, we found 2 of these. And we never found them together, but we found them very, very, very close to each other. And as you can see here, they're extremely similar looking, but there are slight differences between them. And the main differences are in wing formula. You can see here, the one we call chilensis, it's got a very rounded wing, whereas the one we call Heine, it's got a much more pointed wing. And this is actually migratory. This is either resident or a short distance migrant, whereas this is quite a long distance migrant, which explains the difference here. There's also differences in tail pattern, even though they are less clear cut than the difference in wing formula. And on a bird that's sitting on the deck, you can actually see these wing formula differences, although it's not always very easy to be sure of exactly which primaries are visible. So this should be looked at with care, and I think with some experience as well. And in general, you can see that it's got a shorter primary projection here in the chilensis than in the Heine. So based on DNA, we found 4 or perhaps 5 deeply diverged clades. Here's one. Here is another one, a third, and a fourth. And this fourth was actually divided into 2 fairly deep divergences as well. And as you can see, there are song differences. I will play first this one here, Heine, that ranges from eastern Turkey all the way to southern Mongolia. It's got a very scratchy song that begins a bit stuttering and then a long, scratchy song. Then I will play this rufescens, which occurs from Canary Islands all the way across, it's in Spain, and then across North Africa to Western Arabia. Probably going into southern Turkey, where it probably meets this one here. It's got also a rather stuttering beginning of the song and even more scratchy song than this one. Very dry rattling. I will not play this sandlark, but I will play one example here of the eastern, the one that ranges from Western China all the way to northeastern China, adjacent parts of Mongolia and Russia. There is a population which we treat as the same species here in this clade, also in Kazakhstan. We haven't got any sound recordings of the Kazakhstan subspecies, but we got from Western China. But it's got a song that begins much more abruptly than the others, and it's shorter and slightly more melodious. Here. Play that one more time. So differences between the vocalizations here and between DNA. And if we take a lot of measurements on these, they can be seen to form different clusters here in an analysis, in a multivariate analysis. And the sandlark that occurs in the Indian subcontinent mainly, it's fairly similar to these ones from Western China and further east in China, which again separate slightly more. Their song flights and habitats differ also where they come into close contact in Mongolia. So the northern one, the Chilensis, it likes these grassy steppes with fairly tall patches of grass, whereas the other one that is more southerly in distribution, the Haine, which is now called Turkestan short-toothed lark, it likes more sandy soil with these little thorny bushes, caragana bushes. And look at the song flight here. Chilensis, it flies with continuous wing beats when it's singing. There's no gliding here, or extremely short glides. Whereas if you look here at the Haine, when it sings, it glides. So it flaps its wings, otherwise it would drop down to the ground. And then while singing, it extends its wings and it glides throughout the song. So that's quite a clear difference between those. If we summarize these things, we've got this tree here, which shows the divergence between 4 or potentially 5 clades. And here, this black color indicates strong support. A gray color is rather weak support, and white is no support at all. So we got really strong support by mitochondrial cytochrome B, by a multilocus analysis, so that is using mitochondrial and nuclear markers, morphology, plumage and structure, highly supporting of this clade, song as well. In this case, bioclimate, habitat, not so distinctive. So most of these, or actually all these 4, are supported by at least 4 of these traits. Whereas if we compare these 2 clades, which are less deeply divergent, you can see that there's much greater uncertainty. And if we compare these 2 different subspecies of the sandlark, Reital and Adamsi, you can see that there are some distinct differences, but there's also less clear distinctness. And same here if you compare Leucophea and Sibumai, and there's very little difference. So we conclude here there's 4 species, 1, 2, 3, 4. The rufous-napped lark has got a wide distribution, or had a wide distribution until we started splitting them. As you can see here, the rufous-napped lark is the green markings here. Then we've got red-winged lark in this pale reddish-brown color. We've got Somali lark here in very pale brown, and we've got Sharp's lark, which was until quite recently treated as a subspecies of rufous-napped lark as well in Somaliland. And the rufous-napped lark, based on our analysis, we propose is actually 6 different species. Red-winged lark, we suggest is better treated as 2 different species. Somali lark, we want to retain as 1 species, but Ash's lark, which I forgot to say is only known from here, is no longer treated as a distinct species. It's a subspecies of Somali lark. So in this case, there's 9 species instead of 4. And what do we base this on? If we start by looking at a phylogenetic tree, this is based on genomic data. We can see that all of the ones that are that were previously treated as rufous nape lark, highlighted by this white frame here, they do not form a monophyletic group. And here's the sharp lark, which was also until quite recently treated as rufous nape lark. So you see, they do not form a monophyletic group. We've got a monophyletic group here. In one of the analyses, we've got a monophyletic group here. In another analysis, we've got a monophyletic group here. So you see there's differences between different analyses here. But there's also very deep divergences between these. So we've got within the rufous nape lark, we've got 1, 2, 3, 4, 5, and then actually 6 if we include sharp lark, deeply diverged clades. And we also found the one subspecies of red-winged lark to be, we didn't have any genomic data here actually, but based on mitochondrial DNA, it's sister to sharp lark. And as you can see here, ashes lark is not differentiated from Somali lark. So we looked at museum specimens, lots and lots and lots of museum specimens, and we found both plumage and morphometric support for some of these groups. You can see here's an analysis based on morphometrics, so on measurements that we've taken. And we also found in the field differences in the way that some of them raised their crest feathers, a little crest when they were singing, whereas others did not. And also differences in the wing flapping, wing clapping, which some of them do when they sing. So look at this one, what it does soon. There, you could see it flapped its wings very, very rapidly. And you couldn't actually hear the flapping there, but I think you will hear it. I will start by playing the rufous nape lark, the one that we still treat as rufous nape lark, and it sounds like this. I think you're going to hear a little flap there. So a rather short, faint wing clapping just before the song. And then we look at this one here. And now listen. A very, very simple song, but a very strong wing clapping in front of the song. Then we listen to this one here. Very strong wing clapping throughout the song. A very simple song as well. There's another bird in the background there. Then here we've got a species, or well, a type with a simple song, but usually no wing clapping at all. And this one actually has two different song types. The other one sounds like this. So there are differences in song and in wing clapping between different populations here, between different taxa. And here is an analysis based on measurements taken on these. So there's only one taxon here that is not, only one of the genetic clades that is not supported by vocal differences. All the others are. So here's a summary. And as you can see here, again, all the ones with this red asterisk have been treated as rufous nape lark, and we've summarized all the differences here between the different populations. I want to stress that these need to be studied more. sharp's lark, for example, we don't know anything about its vocalizations or behaviors. It's not been seen for 50 years. Kidipuensis is also very poorly known. It occurs in northern Uganda and adjacent parts of Ethiopia. Is poorly known. So a lot more studies are needed on these birds. The bush warbler complex, Locustella sibomai complex, as it was previously known as, is widely distributed in mainland Asia and also on some islands, as you can see here. It has been treated as between 1 and 6 species. And if we look at sonograms, we analyzed songs of all these, and we found some differences. As you can see here, another analysis based on measurements. We've identified some distinct clusters between different populations. And we studied 2 of these in central and southeast China. You can see they look extremely similar. There are morphological differences. This one has a relatively longer tail, shorter wing, shorter bill than this one down here, and songs quite distinctly different. Extremely thin, high-pitched song here, and whereas this one is pretty different, much lower pitched. And it's not only the pitch that differs. You can also see here, if you look at this graph here, you can see that there are other differences as well, not only the pitch that differs. So their distributions are like this. They are mainly parapatrically distributed, but there are some places where both occur together. And we identified some areas in central China, in the Sichuan province, where we found both together. And in most cases, they were segregated by elevation. There's some overlap. And we actually had these 2 individuals were actually caught in the same mist net. This one while playing this song, and this one while playing this song. So they apparently had overlapping territories, but did not respond to each other's songs. And genetically, there are clear differences as well. So you've got a clade here with this one, and you've got a clade here with this one. And the birds in southern Vietnam, for example, which were also treated as the same species, are quite divergent. They also differ in other aspects from the others. So I told you before that I don't trust divergence times very much, but if this is correct, these have been separated for less than a million years, and they seem to be totally reproductively isolated as far as we know, which is unusually fast. And our revised taxonomy suggested there are 6 species, and this one here we described as a new species to science, which we named after China's foremost ornithologist, grand old man, Cheng Chorsin. So here's another example where we have what was previously treated as a single species, the plain-backed thrush, with a distribution from northern Pakistan through the Himalayas and mountain regions of China, central China, and a long-tailed thrush with a fairly similar distribution. We found that there are differences in habitat as well as in vocalizations between different populations. So in the alpine zone here, we've got a song, sounds like this. It's a very scratchy song. Then in the forest, just below, the song is pretty different. much more melodious. So I remember once standing at, I think, 4,100 or 4,200 meters in the alpine zone, hearing this scratchy song right next to me, and then in the forest below, at the same time, hearing this song. Then in central China, the song is also melodious, but even more beautiful than this one here. And the habitat can sometimes be pretty different from here. It can be identical, but it can be more broadleaf forest. The long-tailed thrush has got a pretty poor song, I would say. Anyway, pretty different. So the alpine inhabiting birds, they differ from the two others in all aspects that we studied, and it is sympatric with both of these. So both in central China, it occurs in sympatry with the beautiful singing one that occurs at different elevation, and in the eastern Himalayas as well. Whereas the Himalayan and the Sichuan birds, they differ in all aspects, although less clearly, and we have not found them in sympatry anywhere. They are parapatric. So Pam Rasmussen did an enormously hard work on establishing these morphological differences. She probably measured every single specimen in the world in different museums. And she identified specimens that were clearly belonging to this clade here. And we identified 1, 2, 3, 4 clades here that were quite deeply diverged. And one of these turned out to have new scientific name. So we named it Zoothera Salim Ali, after India's foremost grand old man, Salim Ali. And as you can see on the divergence times, they're pretty similar to divergences or even exceeding divergences between some of the other well-established different species. This one here, we are not quite sure what it is. It's something we have found by genetics based on old museum specimens, and we think we found it in the field, but we need more data. White-browed shortwing, wide distribution across mainland Asia and on Taiwan Island. Differences in plumage between different populations. You can see these are adult males, slight differences in plumage. First-year males, so in the first breeding period, some populations are very similar to adult males, whereas others look just like females. And on Taiwan, all males and females are the same looking. Different songs. Here's this one here. And here's the range of that one. Very peculiar song. And this one, which occurs here, sounds like this. It's a much shorter song. And we actually found them oops. I can't stop it. Okay, that stopped on its own. So when we analyzed measurements of songs, we found clear differences between them. And of course, you can clearly hear these differences. In one place here, we found the two together. They were right next to each other. They did not respond to playback of each other's songs. And based on genetics, they are pretty deeply diverged as well. So we suggested this is actually 3 different species, one here, and one here. So it's become popular in recent years to use DNA to delimit species. And you may say DNA covers everything. Is there any need for integrative taxonomy? Or is DNA-based computer or DNA-based species delimitation sufficient? I would say no, it's not. It's actually, in my opinion, deceptively objective. It can be an important part of integrative taxonomy, but computer-based species limits must be evaluated, in my opinion. And morphological, vocal, behavioral differences are actually very good additional evidence of lineage divergence. They are faster evolving than most genetic markers, and they could show evidence of divergence long before the main part of the genome shows any differences at all. So the genes coding for example, plumage differences, they make up just a tiny, tiny fraction of the genome. So they are totally swamped in these analyses. Also, sexual signals such as plumage, song, sexual displays, they can lead to reproductive isolation long before any genetic differences become evident. Also, if one is using just a few genetic loci, for example, mitochondrial DNA and a few nuclear loci, one of these, in this case, the mitochondrial loci, might provide or will provide a much stronger signal than the others, even though there could be conflict between these different loci. But the mitochondrial signal will be much stronger and will be what determines species delimitation in this case. So my concluding remarks here. Can I tick it or not? Is it a species or is it a subspecies? Does it matter? Well, to a lister, it does. And to legislators, it does. I would say that to me, it doesn't really matter. I think if a certain taxon is distinct, if it's unique in one way or another, it's interesting. I don't really care about whether it's treated as a species or a subspecies, even though when I write a paper or a book, I have to make a decision. But importantly, we need much more research because only a minority of the taxa in the world have been studied in an integrative taxonomic framework. So much more needs to be done. And this needs to be done quickly. It's urgent because subspecies usually has much poorer protection than taxa that are treated as different species. And it's quite possible, we know it has happened actually, that species might go extinct before they become recognized as species. So thanks very much for listening. And I wish to thank all my research collaborators. I have not forgotten any of them, but this rhino is not big enough to list all my collaborators. And there is a list of the cited publications on the last slide I will show here. And same with photographers and illustrators. The names are on the slides. I'm eternally grateful to everybody. And of course, to people, to organizations, foundations who have supported this research over the years. And in case you're interested, here is some further information on if you, in case you want to see more about what I'm doing. Got a webpage here at Uppsala University, ResearchGate, LinkedIn, etc. And if you've got some good papers, even research is a possibility. So here, finally, the literature that I've cited.
Okay. Yeah, there's been some really good questions that have been coming in. I answered a question related to families. Someone had asked if family level taxonomy was any less subjective than species level taxonomy, and I said no. But that did prompt a few more questions. Even if species represent biological entities, aren't they still human constructs to some degree?
Yes, and I would say yes, they are definitely. I mean, for example, when I cycle home, now it's dark, but when I cycle between my house and work, I see a number of different species and they look different. I think everybody would agree that yes, they do exist. They clearly are different entities. But when it comes to things that are not in contact, then, like I've shown some examples of, yeah, then it becomes sort of tricky. But I mean, there was a debate many years ago whether species were real or not. And yes, they are. I would say, yes, they are real, but it depends on, I mean, we can only see what is now. We can look at fossils, for example, but and then it becomes really complicated. Do you draw the line? Just like I showed on this tree that splits, and it's a very gradual process. Where do you draw the line? When is it a species? It's sort of similar, sort of analogous to um, my my development from a baby to a grown up. I mean, where do you draw the line?
Yeah.
When did I become adult? Well, I think probably not yet, but anyway, you could of course say that when you're 18, you're adult. Yeah, but that's subjective. You could just as well say that when you're 25, you're adult, or when you're 65. But anyway, all those gradual, continuous things are subjective.
Yeah. So in your integrated taxonomy framework, you've discussed how you bring in vocal data, ecological data, genetic data. How do you weigh the relative contributions of these various factors in deciding whether to recognize something as a separate species or not?
Yeah, well, that's a good question. So I guess I would say that I weigh them depending on, I don't weigh them, I mean, to me, they're sort of equal. Ecology, habitat is probably, that should be downweighed, I think. But morphology, vocalizations, genetics, all super important. But so then it's up to the level of divergence. How different are they? How strong are the morphological differences? How strong are the vocal differences? How deep is the genetic divergence? Is there any evidence of interbreeding or not? Are they clearly monophyletic? So you have to remember that different lineages will diverge at unequal rates. So you might have, I mean, you can think of us actually. You can think of humans and compare to our closest relatives, the chimpanzees, and compare the chimpanzees morphologically to the other great apes. We have diverged enormously in appearance, whereas the other great apes have not diverged so much. So you have to remember that certain traits, certain lineages will diverge quite dramatically, whereas others will not diverge so much. And you've got the leaf warblers, for example, that are very similar in appearance, and the bush warblers, whereas the vocalizations are usually much more divergent. But in some cases, you have even leaf warblers that have very similar vocalizations that are quite distantly related and that have overlapping sympatric distributions, but they sound very similar. Maybe not to them, but to human ears, they sound very similar. But the birds themselves, I'm absolutely sure, have no problem at all to distinguish. And I think I can distinguish between them. But I know lots of people who say, oh, they just sound the same to me. But
Yeah.
so there's a lot of variation in nature, and that's what makes it so incredibly fascinating.
Yeah. So we have two kind of related questions from students, a student and an early career ornithologist. So the first asks for tips and recommendations for making a career out of taxonomy. And the second from the early career ornithologist asks, what skills would you recommend to focus and improve for kind of long-term benefits in the field of ornithology and taxonomy?
I think all ornithologists today, I mean, especially if they're interested in taxonomy or systematics, they need to develop bioinformatics skills. That is really crucial because there's going to be more and more studies of DNA in the future, and even if you're not focusing on taxonomy systematics, DNA is nevertheless extremely important. But then, of course, I think you really should try to do fieldwork as well. I think it's super important. If you only know DNA, if your only skill is in bioinformatics and you don't know the birds, you don't understand birds, that's not so good. You really need to be able to study the birds in the field, see what they're doing, see how they're behaving, study their ecological characteristics. And I think if you haven't got that ability or interest, you're not going to be as good as you would be otherwise.
Yeah. When there are undescribed forms, what can folks as amateurs and birders do to help them get recognized as formal taxa?
Well, I mean, even amateur birders could describe new species. There's no, it's nothing that only professional ornithologists can do. But you need to follow certain rules in order to describe a new species, and that can be really complicated sometimes. But when a birder travels to a place and finds something that they think might be a new species, the important thing is to try and document it in as many ways as possible. Try to get photos, try to get video, try to get sound recordings, study its behavior, and then notify others about its existence so that others can go there and study it and get more data. And in order to describe a new species, one actually needs to have a type specimen, which means collecting a specimen. And sometimes it's possible that a specimen might already be available in a museum. So it's just been overlooked. So for example, with the Himalayan thrush that I mentioned, Pam Rasmussen, who was heavily involved in that study, she looked at, I don't know, maybe thousands of specimens in different museum collections, and she found multiple individuals of this Himalayan thrush that had just been overlooked in the museum collections. Nobody had realized that they were distinctive. So in many cases, there are things available in collections that people just have overlooked.
Yeah, totally. Let's see. Laura, how are we doing on time?
Let's see, we still have 270 people. So I'd say,
well, I'll pick another question, and if you're up for another question, Per.
I can stay for another hour. Yeah.
Okay, some people are saying we don't want to leave, so we don't often have such expertise here. So please go right ahead.
All right.
Just carry on.
So this is an interesting question that came in. Were all the so when you were comparing the vocalizations of the warblers in your various studies, were all of these vocalizations recorded in natural areas away from urban or industrial noise? How might that affect their song? And can urban noise pollution alter these features enough to kind of affect not only identification, but potentially like speciation as well?
Yeah, well, I think all of the species that I've sound recorded have been in areas. I mean, sometimes I've got, of course, human pollution in the background. Of course, it's unavoidable in many places. But I think the risk that, I mean, it has been shown in certain studies that birds can change their frequency if they live in a very noisy habitat. like in a city, they can increase their pitch, their frequency to be heard more clearly. But still, it's the same song. It doesn't really change that much. And
Okay.
I mean, and these populations will nevertheless be in contact with other populations. So there will be sort of, as noise will diminish as you move away from the city, the songs will also change back again to sort of the normal. So I don't think humans will create in a species just through noise pollution. No.
All right. So how does the speed of an adaptive radiation, how might that affect some of the various characteristics that you use for species delimitation? For example, might behavior or song change more rapidly in an adaptive radiation than other features, or vice versa?
Oh, well, that's a question I really can't answer. I mean, adaptive radiations in general are considered radiations where uh, different species evolve from a single species, evolve into multiple species that differ in ecological niche and quite substantially in structure. So they feed on different things. You've got the vangas on Madagascar, for example. You've got the radiation on the Darwin's finches, and so on and so forth. And it's usually considered to be feeding related differences.
Right.
And feeding related differences can, of course, be used um, to be important for mate choice, which then could be important for reproductive isolation. But I think with adaptive radiations of that magnitude, they will be recognized as species long before they have diverged to the level where we talk about an adaptive radiation. I mean, you could talk about the yellow wagtails that I showed as a radiation, but it's
Yeah.
not an adaptive radiation because they
Right.
occur in the same habitats, they all feed on the same things. But I would nevertheless say that it's a kind of radiation because they have diverged so much in plumage in lots of different geographical areas. And in this case, in this particular case, I think it's sexual selection. So females have selected for these beautiful male plumages, and there's been for various probably charms, for various stochastic reasons, there's been different selection in different areas, and they've been geographically separated probably. And yeah, anyway, they certainly have diverged in different
Yeah.
plumage. And that's something we've been working on for a long time. And yeah, we'll see if we can finish at least one paper this year, hopefully two or more. Yeah.
So how many lines of evidence would you either need or like to have to consider an integrative approach?
Oh, that's another good question. I don't want to I don't want to say a specific number. I mean, the more the better, I would say. So I didn't show a slide of yellow-billed and black-billed magpie. I had a slide of those, but the magpies we found quite a few years ago, based on mitochondrial DNA only, that there are 6 main lineages that are pretty divergent between, I think, 1.4 and 3.5 million years. But there is one very shallow divergence, and that is between the yellow-billed and the black-billed magpie in the US. They're really, really, really similar genetically, based on mitochondrial DNA. To the best of my knowledge, nobody's studied their genomics yet. That would be really
Yeah, I
interesting.
don't think so. Yeah.
Yeah, yeah. And these are parapatrically distributed. They don't come into contact. So how do you treat them? I mean, genetically, based on genetics, no, you can't really say they're different species, even though they form monophyletic groups, but nevertheless, their divergence is so incredibly shallow that would you treat them as different species or not? Well, it's a matter of taste, really. but based on their phenotypes, based on this bright yellow bill of the yellow-billed magpie, yeah, I think most people want to treat them as different species anyway. So it's tricky. It's tricky
Yeah.
sometimes, and I don't want to say that you need like 3 or 4 or 5 or more traits to yeah.
Yeah. Let's see. So you kind of have addressed this a little bit in different aspects of your talk. So, looking at all the recent changes in taxonomy, we've seen a lot of different taxa that have gotten split. You've showed that like beautiful steady increase in the number of species over time. But some species also have gotten lumped over that same time period. What are some of the compromises that we might potentially need to make when we just can't fit, necessarily what's happening in these populations into this our human constructed species concept?
Mm. Yeah,
At what point, yeah, at what point do you just have to like make a decision and
yeah. I mean, that's what all the different taxonomic committees are struggling with. We have to make
yeah,
decisions because
exactly.
we have to say, is it a species or is it a subspecies? We have to make a decision, even though we may agree that we can't really make an objective decision. It has to be subjective, and we just have to, based on the evidence available, we have to make a decision. Just
Yeah,
because, just because we have to, and uh, and importantly, if we lump 2 species into 1 species, and then with time we realize that we get more data, and we realize, oh, they're actually distinctive. Let's split them again. So it's not stable. Taxonomy is not stable. I mean, in some cases, it's surely stable. Great tit
sure.
and blue tit will never be lumped. But the different blue tits, well, who knows? Maybe somebody in the future will realize that those genetic data were, yeah, not trustworthy after all. I doubt it, but anyway, things,
Yeah.
yeah.
All right. There is, there's a number of questions on like very specific taxa that you didn't necessarily cover in your talk. I don't know. Um, People are curious on your thoughts on red poles,
Okay,
a very
okay.
complicated
Yeah, yeah.
system.
So I don't really have a strong opinion on red poles. They clearly are very closely related, very similar, and um, I think most people today consider them a single species, the red pole. Whereas previously, so I think in Sweden there's never been more than 2 species recognized by the Swedish Taxonomic Committee. So the hoary red pole and the common red pole. Whereas others have recognized also the, what's it called, mealy red pole, the southern
Yeah,
continent.
the lesser. Yeah.
Lesser red pole, yeah, lesser red pole. Sorry, lesser red pole. Um, and I mean, they're clearly very, very, very closely related. And I know it has been shown that the main difference between the hoary and the common is in an inversion on one of the chromosomes, the first chromosome. There's an inversion in which there are genes coding for both plumage differences and the size of the bill, which differs between those. And apart from this inversion, they're basically the same thing.
Yeah.
But I also know people who claim that, at least in northern Scandinavia, they mate assortatively. So hoary mate with hoary, common with common, and they are ecologically at least moderately well separated. So, like I said before, differences in mate choice can evolve much faster than genetic differences. So,
Yeah.
I mean, in this case, it's been shown that there is a lot of interbreeding, there is a lot of introgression on the genomic level, but maybe in certain areas they don't interbreed very much. Maybe in other areas they interbreed more, and so on. So it's with evolution, it's really hard to draw firm conclusions many times, and we
Yeah.
all
So
want to put things in neat boxes, but we have to accept that we can't do that. Sometimes we just have to say, okay, we don't
this is
know.
our best guess and
Yeah,
move on.
exactly. Move on and be happy anyway. Enjoy them anyway.
Yes.
I mean,
Yep.
I love hoary redpolls, even no matter whether they're treated as species or subspecies. They're beautiful birds. I love watching
Yep.
them. It's been a long time, but since I saw one. But anyway, yeah.
Yeah, same here. All right. I think that covers a lot of the big questions. So I think in the interest of kind of where we're at with time, I think we might, if you're yeah, I don't know. There's a question, I'm sorry, I'm just scrolling through. There's a lot of oh, here's a fun question. In when you're talking about dating things based on genetic evidence, how was the date of those splits estimated? Always a fun,
Oh, that
fun.
varies. That varies.
Yeah,
I mean, the best way of doing it is using fossils as calibrations. But when it comes to closely related species, there usually aren't any fossils that can be used. So then it's usually molecular clock that's been used. And yeah, like I said, I don't really trust um, time estimates per se. Relative divergence times are more interesting. So if you have really deep splits in a phylogeny, that is more interesting than whether you say that it's 2 million years or 5 million years or
yeah,
so. But then you have to take into account that also relative divergences can be affected by various processes such as introgression, or yeah, lots of things can affect that. So one always needs to be careful, and that's why it's so important to take as much data into account as possible. Don't trust just a single class of data. Use as
yeah,
much as you can. So that's the key of integrative taxonomy. Use as
yeah,
many
and
lines of evidence as possible.
yeah, you showed just even comparing divergence between mitochondrial DNA and nuclear DNA, just like wildly different
Hmm.
patterns and estimates. So,
Yep,
yep,
indeed. I mean,
multiple lines of evidence.
if you have a mitochondrial divergence that is deep, and you have a nuclear divergence showing a similarly deep divergence for the same taxa, fine, that's good
Yep,
evidence. But if you have conflict, if you have like deep mitochondrial divergence and poor nuclear divergence, or the opposite, which is less common, then yeah, you need to consider
yeah,
other things as well.
yeah. All right. I think this might be a good place to wrap up. Um, thank you so much, Per, for your time and expertise. Thank you, everyone, for coming.